Welcome to G Fun Facts Online. Today, we're cracking open a golden window into a lost world. We're talking about amber. This beautiful gem is more than just jewelry. It's a time capsule, freezing moments from millions of years ago in perfect, three-dimensional detail. Inside these golden tombs, we find not just creatures, but entire ecosystems, from the age of dinosaurs to the rise of mammals. Forget dusty bones in a museum. Amber shows us prehistoric life as it truly was. Vibrant, dynamic, and often very, very sticky. So how does a simple drop of tree goo become a portal to the past? It's a journey that takes millions of years. It all starts with resin, not sap. Sap is the watery lifeblood of a tree, like our circulatory system. Resin is the tree's defense mechanism. When a tree gets injured, it bleeds this thick, aromatic resin to seal the wound, repelling insects and disease. For this resin to have a shot at immortality, it needs a special chemical makeup, rich in compounds called terpenoids, which resist decay. Most resins just break down, but these special ones can last. The transformation happens in two main stages. First, over thousands of years, the resin's molecules link together, forming larger, more stable structures. This process, called polymerization, turns the resin into a substance called copal. Copal is like a proto-amber. It's younger, softer, and can still be dissolved by solvents like acetone. It's a teenager on its way to adulthood. For copal to become true amber, it needs to be buried under layers of sediment, away from oxygen. Over millions of years, the immense pressure and geothermal heat from the Earth cook it. This maturation drives off any remaining volatile compounds and causes the polymer chains to cross-link even further, creating the hard, inert, stable substance we know as amber. The world's most famous amber deposits each open a door to a different era. Baltic amber, from 40 to 60 million years ago, gives us a look into the Eocene epoch. Dominican amber, famous for its clarity, is about 15 to 20 million years old from the Miocene. And the holy grail for many, Burmese amber, or Burmite, is a staggering 99 million years old, offering a direct view into a forest from the Cretaceous period, when dinosaurs still walked the earth. The real magic of amber is what it traps inside. The regular fossil record is biased towards hard parts, bones and shells. Amber, on the other hand, preserves the small, the delicate, and the soft-bodied. It's a microscopic view of ancient forests. By far, the most common residents are arthropods, insects, spiders, and their relatives. Flies, ants, beetles, and wasps are found in staggering numbers, often preserved down to the finest hairs on their legs. In Burmese amber, scientists found a 100 million-year-old scale insect, frozen, with over 60 eggs on her back, the earliest direct evidence we have of insect brood care. Spiders are another common find, sometimes with their webs intact, or even caught in the middle of an attack. One incredible piece of Burmese amber shows a 100 million-year-old spider attacking a wasp caught in its web, a moment of predation frozen for eternity. But it's not just insects. While vertebrates are much rarer finds, they are scientifically priceless. Tiny lizards and frogs have been found, perfectly preserved with skin, scales, and even color patterns. One of the most electrifying discoveries ever came from Burmese amber in 2016. A piece originally meant for jewelry was found to contain a 99 million-year-old dinosaur tail. This wasn't just a feather. It was a segment of tail with bones, soft tissue, and feathers all intact. It belonged to a small, bird-like dinosaur, a juvenile coelurosaur. This was the first time skeletal dinosaur material was found in amber, providing undeniable proof that many dinosaurs had feathers. The feathers themselves were more downy and simple than those of modern birds, likely used for insulation or display, not flight. Isolated feathers are more common, showing the whole evolutionary journey from simple filaments to complex flight feathers. We've even found ancient mammalian hair, direct evidence of our furry ancestors scurrying through the dinosaur-era forests. And let's not forget the plants. Amber preserves the ancient botanical garden that the animals lived in. We found 100-million-year-old flowers in the very act of sexual reproduction, with pollen tubes penetrating the stigma. We found the sticky tentacles of 40-million-year-old carnivorous plants, relatives of a species now found only in South Africa, showing they were once much more widespread. Mosses, ferns, and fungi help paint a complete picture of the ancient forest floor. Amber's preservation even extends to the microscopic world. Some of the most bizarre finds are so-called zombie fungi. In Burmese amber, these ancient parasitic fungi, relatives of the modern Ophiocordyceps, are seen erupting from the bodies of ants. This shows that this gruesome form of mind control, where a fungus takes over an insect's body, was already happening 99 million years ago. By studying all these inclusions together, scientists can reconstruct these lost worlds in incredible detail. Take the 99-million-year-old Burmese amber forest. It was a dynamic coastal environment. The proof? An ammonite, a shelled marine creature, was found trapped in the same piece of amber as forest-dwelling mites and spiders. This tells us the resin-producing trees grew right next to the sea. This forest was a complex ecosystem filled with predation, parasitism, and the coevolution of the earliest flowering plants and their pollinators. And, of course, small, feathered dinosaurs were an integral part of this world. Now let's jump forward in time to the Baltic amber forest, around 40 million years ago. This was a warm, temperate world, much like modern-day East Asia, not the cold northern Europe of today. The forest was a mosaic of habitats, from coastal swamps to inland forests dominated by conifers, oaks, and magnolias. The sheer number of certain insects, like aphids, allows scientists to deduce seasonal patterns, suggesting that most resin flowed in the spring and summer. Finally, let's visit the tropical paradise of Dominican amber from about 20 million years ago. Its amazing clarity gives us the most detailed look at any prehistoric tropical forest. It was a humid, mountainous jungle, teeming with social insects like stingless bees and termites, giving us a wealth of information on the evolution of their complex societies. Scenes of combat, like an assassin bug grappling with a bee, bring the dynamics of this Miocene ecosystem to life. So how do scientists get all this information out, especially from amber that's cloudy or opaque? This is where the modern scientist toolkit comes in. For decades, fossils in opaque amber were a hidden treasure, but now, techniques like high-resolution X-ray micro-CT scanning allow scientists to create a detailed 3D model of the inclusion without ever touching it. They can digitally dissect a tiny insect, revealing internal structures crucial for identification. For the toughest cases, they use synchrotrons. These massive particle accelerators produce X-ray beams billions of times brighter than hospital X-rays, allowing them to see through completely opaque amber. They can then 3D print these digital models, creating perfect physical replicas for hands-on study. The amber itself is also a chemical archive. A technique called Fourier transform infrared spectroscopy, or FTIR, shines infrared light through a sample to get a unique chemical fingerprint. This can tell us the type of tree the resin came from and can easily spot fakes. Now we have to talk about the elephant in the room, or rather the dinosaur in the amber. Can we pull a Jurassic Park and clone dinosaurs from DNA found in blood-sucking insects? In the 90s, some studies claim to have extracted ancient DNA from amber, causing a wave of excitement. It seemed science fiction was becoming science fact. However, the scientific consensus today is a firm no. The problem is that DNA is an incredibly fragile molecule. After an organism dies, its DNA begins to break down immediately. Studies have shown that DNA has a half-life of about 521 years. This means that even under the most perfect preservation conditions, every DNA bond would be destroyed after about 6.8 million years. The dinosaurs died out 66 million years ago. Their DNA is long, long gone. So what about those early, promising results? It's now widely believed they were the result of contamination. The methods used to detect tiny amounts of DNA are so sensitive. They can easily pick up modern DNA from the air, from fungus, or even from the researchers themselves. So while amber is our best window into the appearance and ecology of ancient life, the genetic blueprint for a T-Rex remains lost to time. It's also important to remember the amber fossil record has its biases. It heavily favors small terrestrial creatures that lived on or near the resin-producing trees. A big animal could just pull itself free. Aquatic organisms are naturally absent. It's a window, but a window that looks out onto a very specific part of the ancient landscape. Amber is a fundamental key to understanding the evolution of life on Earth. Each inclusion tells a story, a character in a grand ecological narrative spanning millions of years. It gives us a tangible connection to the deep past, preserving moments of life and death with a clarity no other fossil can match. As technology advances, we're entering a golden age of discovery, decoding secrets that have been locked away for eons. The dream of resurrecting dinosaurs might be fiction, but the reality of what we can learn from amber is even more profound. Thank you for joining us on Fun Facts Online.